So it was around this time of the year when I first started my worm culture and when I began grad school. So today I'm excited to share and bring you along on this 4-year journey to the world of the physically entangled, focusing on the collective behavior of aquatic worms or worm blobs. And aside from collective behavior, I also explored diverse fields such as soft, active, and physical and entangled matter, living polymers, topology, and even soft robotics. Now, collective behavior occurs when groups of individuals move in coordinated ways without a leader, like starling birds during murmuration, as seen here, or crowds of people, such as in the mosque at Mecca. This relates to active matter where self-driven individuals interacts with others, creating emergent behavior. Now, emergence occurs when simple rules combine to form something much more complex. And this type of autonomous and decentralized coordination inspires fields like swarm robots, where cheap robots could operate with minimal human input. However, what isn't swarm robotics is like these robots that we can see here in during the Tokyo Olympics of 2020. Now, these drones are individually pre-programmed, and they could move to a position that is determined by a human being, which lacks autonomy. What is swarm robotics are like these kilobots down here, which are cheap, autonomous, and decentralized units equipped with vibration motors and infrared communication that follow basic rules, such as go move towards a robot. And this allows for emergent shapes to occur without having to program it in advanced ways. Now, robots inspired by worms add the active polymer part into it. Just as polymers consist of individual units called monomers, the segments of worms could be considered as monomer, such as this toy over here, and this increased length adds shape-shifting capabilities valuable for soft robotics. These slender and adaptable robots are useful in applications such as in medicine, like endoscopes, search and rescue, and for example, origami-inspired robots, as shown here, that can navigate and move around obstacles to hit its target. In fiction, soft, squishy, and shape-shifting objects have been popularized in media, typically in a terrifying manner. The closest thing to the worm blobs is from the Netflix version of of the game called Halo, where one type of superorganism called Lekgolos can grab humans, objects, and move through all kinds of terrains, which is the worm here, in an entangled matter. She's my hero, by the way, a literal worm blob whisperer, actually depicted in the movies. Of course, I'm just kidding. It is important to note that the worms I work with are quite gentle and not dangerous, and they just stink sometimes. We classify entangled matter according to their aspect ratio, as seen over here, which is length over diameter, and the complexity of their topology, which uh, and we do that by calculating the linking number. And that is a mathematical equation that can be thought of as a measure of the number of braids between two curves. And these are some of the real examples in passive, active, and robotic cases. And more information could be found in our review paper published in Soft Matter last year. So something that has high aspect ratio but low linking number could be the sticks on a bird's nest. And you could still mold around this to make a shelter. If we reduce the aspect ratio, rigid U-shaped particles like staples only form single braids before breaking or deforming, but they can still show emergent dynamics, as seen here, being capable of picked up by a pencil. If we reduce our particle, let's say, now let's increase topological complexity by making the arms move around. Now we get an example in biology, like ants in an ant raft, which is a 2D shaped raft that can float around collectively and migrate in case of flooding. And in Professor Goldman's lab, they have smarticles, which are three-linked robots that can flex its arms around. And we have shown that this is capable of collectively locomoting towards a light source. And due to their flexibility and large aspect ratio, worm-like filaments, or our living spaghetti here, can display complex emergent behavior and dynamics. Over 4 years, I published 7 papers denoted in white text, 5 of which I've first authored and co-first authored, and works in yellow denotes that works that are still in progress with the buoy that is almost completed. This review paper here summarizes our findings from these publications shown in white. The 3 main aims of my entire research can be best summarized by this video. In aim 1, I looked at why worms blob in their abiotic and biotic factors, specifically targeting oxygenation and predator-prey interactions. In aim 2, I look at the topology of how worms entangle and untangle using ultrasound and high-speed tracking, as well as look into the physics of their internal mechanical stress from using experimentation and simulation. In aim 3, I use what I learned from previous aims to describe complex emergent dynamics, which is, for example, how worms collect and gather and consolidate objects around them And this behavior, which I discovered called the worm buoy, where worms use surface tension to suspend themselves collectively on the interface. And it's going to take me several hours to go through this. Therefore, to respect everyone's time, I will be covering my most significant work to include topology, collecting, gathering, worm buoy, where I will be taking results from the other papers to support my data. I'd like to give a brief moment to cover our organism's biology. Most of this information came from the late Dr. Charles Drews, who I'm going to refer to as Charlie. He passed away in 2004 and was a passionate teacher that was a pioneer of using black worms in classrooms. These worms are from the phylum Annelida. Their class is from oligochaete. Oligo means few, and then chaete means hairs, because they have little tiny hairs that can protrude out for attraction. And other oligochaetes includes the earthworms you find in your gardens. Its scientific name is Lumbriculus variegatus, and they're commonly known as California blackworms. They are 0.5 millimeter in size and 2 to 4 centimeters in length, but they can grow all the way up to 10 centimeters in the wild. Worms typically crawl around using peristalsis as seen with earthworms, but however, blackworms are unique in that they can also swim, but only for short durations. And these results are from Charlie here. So if you poke the anterior part of the worms or the head part, they do something called a body reversal. So they flip their heads and tails and swap in places. And soon after, they'll do something called a helical swimming, swimming from a 3D direction of whatever direction they want. And that occurs when you poke them in their posterior section. So why do worms blob in the first place? When I first came into the lab, Dr. Oscar Aiden, now a professor at Notre Dame, investigated the worms by taking them outside of their water and observing how they react. And after a few hours, the worms blob up into a spear. And she found that they do this in order to prevent themselves from drying out or desiccating. So after a few hours, the outer layer tends to be dried up and dead where the inside layers are protected from drying out. Many worm scientists or enelidologists hypothesize that worms blob due to thigmotaxis. Thigmo means touch, taxis is movement. It is thought that the worms feel safe being hugged all around as they like to burrow in sediments as well as entangle with one another. I started my first worm culture in October 2020, ordering them from commercial worm farms as they are cheap and easy to obtain. And here the worms are eating on some fish pellets, which is what we feed them. I also like to watch worms eat and how they manipulate their food around, which actually foreshadows one of the projects that I have I'll show later. The Worm Blob team has expanded, and now there's around 10 to 12 people in a given semester with tens of thousands of worms in the lab. And you can actually see the aquarium, which is back there. When I first started my first worm culture, I was reading through Yasmin's paper, and I was really curious to see what her results are in the paper. She discussed that worms are negatively phototactic, meaning that they don't like bright lights, and they tend to compress when you blast them with really high intensity light. So I found this UV light in the lab, and out of curiosity, I pointed it at the worm blob. Suddenly, this highly entangled blob blew up in all directions, which both mesmerized and freaked me out as I thought the worms were actually going to jump out of the water. This also occurs when I feed worms, and it's raining fish flakes now, which scares the worms. It's been hypothesized by Charlie that this is a predatory escape response at the individual level and can collectively confuse a predator, making it difficult for them to catch a prey after being mesmerized, similar to a lion hunting zebras. They move so quickly that I need to use a high-speed camera, and here I'm capturing them at 1,000 frames per second using an electrical stimulation from a 9-volt battery, which doesn't hurt the worms. It scares them. And here, observe the elegant dance of the worms as they do their helical swimming, where they make little tiny loops, which is important for later's explanation of how they disentangle. This is surprising because worms take significantly longer to entangle with one another. And here you can see the timing here. I had to speed up this video in order for you to see their entanglement. Yet it takes them such a short amount of time to untangle. So what's going on here? With that, we started a collaboration with some mathematicians in MIT, and this is Vishal Patil, who is now a professor at University of California, San Diego, and he was working at that time with Professor Jorn Dunkel, who is still at MIT. And this is part 3 of my work in the topology section of my thesis. The central question we aim to answer is, how do worm blobs untangle so quickly? And to answer that, we have to first answer how do worm blobs tangle up in the first place. As the primary experimentalist on this study, I initially faced significant challenges in visualizing the inner structure of a lime worm blob. I explored various imaging techniques, including X-ray, confocal microscopy, fluorescently labeling the worms when I tried labeling worms with dyes to track their movement within the blob, and even photoacoustic tomography. Ultimately, what worked was ultrasound, a method commonly used in pregnancy imaging. And we collaborated with David, who was a grad student at Dr. Stanislav Emilianov, Professor Emilianov's lab from Georgia Tech, to perform this imaging. And Emily and I, Emily being an undergrad student that was working with me at that time, prepared the sample using gelatin, and it has to be at the proper temperature or worms will disentangle and we'd lose the dynamics. And of note, we tried to immobilize the worms and keep them kind of placed in there. We used gelatin, and we didn't want to anesthetize them because that would cause the structure to relax and we would lose the dynamics we're trying to capture. And once the ultrasound data was collected, it was passed to Tuhin, shown here, who handled the segmentation and manifold learning. A technique used for simplifying and organizing the complex 3D data. And part of that is to label a worm from head to tail, and hopefully you don't end up with 3 heads or 3 tails at the end of the segmentation. If not, you have to start over again. Finally, the data went to Vishal for detailed topological analysis. To analyze the internal topology of the worm blob, we used the ultrasound data to calculate the linking number, or LK, which essentially tells us how many times 2 worms are braiding or twisting with one another. The linking number is commonly applied to closed curves, like rings. For example, when the linking number is 0, 2 rings are simply touching or sitting next to each other without intertwining, where an LK of 1 indicates that 1 ring is looping through the other ring. Now, for open curved worm system, we modified this equation but the linking number is a little different here. When the linking number is less than 1/2, that means the worms are just touching each other. And if it's and then higher values indicate an increasing level of braiding between the worms, higher than 1/2, showing us where and how complex these internal tangles could be forming. From that, we created a contact map where each worm is represented as a point, and they're connected by these lines, which shows how they're touching one another. We found that each worms are braided on average with 2 others. And this level of connection, eventually leading to the tangling index, helps the blob stay together as a cohesive structure while allowing them to remain at a critical enough point to untangle quickly. And this balance between structure and flexibility is key to the entangling and untangling behavior, which we'll see next. And I won't have time to cover what the tangling index means, but it has a far more richer meaning to it, including a puzzle in math called the picture hanging puzzle. And if you're interested in the technical details, more information could be found in our paper as shown down here. Note the timescale of the dynamics here between the tangling and untangling. For complex head dynamics, we manually track the head motion of population of worms across multiple trials. And again, we did this by manually plotting the head of the worm, putting a dot over it. And this was quite complicated because worms tend to go up and down each other, and they rotate as well, making this a very tedious approach. And between Emily and I, we tracked over 45,000 points over 3 months. And here's the helical motion of the worm again, which the worm can turn both direction, left or right. Starting at the head of the worm, we define the worm is moving at a velocity v and is turning at an angle theta. The rate of theta is alpha, or the turning speed, which represents angular velocity, which is how quickly the worm rotates in one direction. Next, we define lambda, which represents the switching speed, or how quickly the worm alternates from left to right turns. Now, if we divide these 2 terms, we get the dimensionless number called chirality number. When the chirality number is high or greater than 1, worms tend to move in a consistent direction, like a corkscrew. With lower chirality values, the worm frequently switches direction, creating this figure 8-like path. Now, with this basic steps or move sets, let's see how they behave in simulations using a Kirchhoff model with 100 worms, and this work was done by Vishal. Here we observe how chirality impacts the entanglement. Observe that when the chirality number is low, where the worms are frequently switching directions, it leads to the worms untangling, whereas a chirality number that is high, that leads to the stable formation of blobs as worms are braiding around each other by corkscrewing, as shown in this toy here as well. So they turn into one direction, they turn to braid like this. This leads to a stable tangled blob structure. In this work, we explored the entanglement dynamics of worm blobs, from ultrasound imaging to experiments to simulation, recreating their intricate movements. And these results are significant because they reveal how simple organisms achieve complex entanglement and disentanglement or untangling dynamics through basic motion patterns. Understanding these principles have broader implications across scales, from the folding and unfolding mechanisms in DNA and protein to larger systems where entangled behavior needs to be controlled, like in fiber or polymer networks, and of course in swarm robotics, where its basic rules, again illustrated in this comic by Jordan, says that for worms to entangle, they need to do the corkscrew. So everyone do the corkscrew, slowly twist around and round your partner. And if they want to disentangle, quickly do the figure eight, left and right and left and right, and that's how they that's how they untangle with one another. And of note, this is Alexander the Great, and if you're interested to know why he's in this comic, feel free to visit our website to check out the rest of the comic, which is also translated in multiple languages. Now let's go back to the fluorescing gummy worms. Again, these vibrant colors was a byproduct of my attempt during the topological studies, which didn't work as intended for tracking purposes, but it did lead to an unexpected discovery. The green color comes from non-toxic fluorescein, an organic compound that grows green under certain light, and the orange is rhodamine. So I placed a few worms in a petri dish with fluorescein to label them, and fluorescein looks like a powder when it's dry, and once you put them in water, they dissolve out, leaving a suspension with some of the particles underneath, as it's denser than water. But out of curiosity, I wondered how did it end up into a singular clump. So I took a pipette and mixed up the petri dish and hit the record button. So it turns out that worms can actively gather materials and pack it together, where smaller clumps are weaved together. So I started a simple question here: how do worm blobs gather materials around them? And to tackle that, we need to answer, how does a single worm even do it in the first place? This is part of aim 3 of my work, which is the complex emergent dynamics. In freshwater ecology, functional feeding groups classify organisms by their feeding role. Shredders chew on plants or decaying material, consuming larger particles like caddisflies. Collectors, like blackworms, gather smaller particles from sediments, and there are also filterers that can filter out particles from the water. Scrapers remove organic material from surfaces like snails, and then predators consume other organisms or use piercing mouthpiece like a proboscis, which is like leeches. This foreshadows another project I'll discuss later. Microscope observation reveals an invisible force around the worm that tracks the object, such as pieces from an acrylic mesh. And that's actually mucus, which worms secrete and use for lubrication. But here it also helps them move a particle around. They also use a hook shape on their tails as a collection point. So fluorescein and acrylic plastics are not typically found in their environment, or I hope not. But one common soft material that is found in their environment and also in our rearing is algae, which the worms eat using an aversible pharynx. And aversible pharynx means they can evert or throw their throat out, and they use that to chomp on the algae. With that, we used algae as our material for the experiment. The study was done in collaboration with Dr. Nguyen, who was a postdoc with Professor Peleg at the University of Colorado, Ishan Tiwari, who is a postdoc in the Bamla lab, and 3 undergrads, Emily, Jessica, and Darshan. So here we observe a single worm under a microscope using 10 milligrams of initially well-mixed algae. Watch how the worm elegantly clumps the soft material into a singular clump, seemingly creating some sort of an edible shelter out of it. Worms employ some movesets, like here it shows a peristaltic aggregation, where the worm uses peristalsis to ratchet the clump of algae from its head down to its hook tail. And we have worms gather other materials as well, employing other movesets, and more information could be found in our paper, just noted down there. But are these movesets, or even the mucus, actually important to collect materials? And to understand the mechanism more in depth, we aim to extract the underlying rules by creating an active polymer simulation. Here we represent the worm as a self-propelled active polymer subject to interaction potentials like spring bending and modified Leonard-Jones type, or van der Waals. More information could be found in our ICB paper or in our Frontiers paper, where it originally came from. From this simulation, we introduced 2 additional forces, epsilon WP and epsilon PP, which represents the attractive force because of the mucus, where WP represents worm particle and PP represents particle. The main takeaway here is that short-range attraction provided by the mucus helps particles to clump and collect effectively around the worm. I would like so as the interaction forces get stronger, it ends up being a clump where the worm gets kind of stuck in one place. So I would like to point out that even with minimal attractive forces, reflecting how dense particles like sand, which mucus doesn't affect too much, can still be clustered together effectively as worms push it all around. This serves as motivation for a follow-up project. In this project, we show that robotic systems made of interconnected vibrating units called bristlebots, named after and about the size of a toothbrush head, can be validated through simulations and replicate the worm's clustering on a larger scale using hard particles like sand. In the absence of explicit attractive force, the simulation of robotic polymer uses flexibility and an active head to gather particles autonomously. And this work is co-led by Rosa, who is a graduate student with Professor De Blay from University of Amsterdam, as well as Pratusha, who is a research scientist in the Baumla lab. Overall, the collect and gathering story reveals that flexibility and activity governs the efficiency of particle clustering, and that this principle applies universally across biological, robotic, and simulated filaments across various lengths. These findings paved the way for designing adaptable software robots capable of autonomously performing tasks like particle collection. So stay tuned for this work to be published in the next few months. In earlier slides, I mentioned that out of curiosity, I watch how worms eat their food and manipulate it all around, sometimes to study how they chemotax or move towards food cues as well, like as shown here when I stumbled upon something completely unexpected. Honestly, it's moments like these, just playing around in the lab and testing small things, and then that ends up leading to some of the most interesting discoveries. So I walked into the lab one day and noticed something completely wild. Again, the worms are supposed to be feeding on the ground here, but here they're floating um, with their tails seemingly on the interface and their heads on the bottom. And they are denser than water, so they typically sink as soon as you put it on water. And if you look closely down on the heads part, they're actually still eating some food here using their reversible pharynx, which you'll see here. And they're also capable of manipulating food around if you watch them close enough. Here, the worm is actually picking up the food as seen there. They actually use their reversible pharynx like a mini suction cup and can pull objects more than 3 to 4 times their body size, as discovered by Yasmin. In fact, they use their pharynx to locomote and drag the buoy structure around using the nearly frictionless air-water interface. Here's another view captured on my phone in a more spherical shape, which is how I named this the worm buoy. With that, I asked the question, what is the function of this worm buoy and how does it even form? This is part 3 of my work in the complex emergent dynamics. Let's take a look at what other organisms use to interface. In comparative animal behavior, many organisms use the air and water interface to survive. For the bottom side here, we have mosquito larvae. They have a siphon, which they use to breathe and hang down via surface tension-driven mechanism. Next is the aquatic beetle, a buoyancy-dominated system with bubbles on their abdomen and on their legs that pushes them up on the interface. And they do this by hiding on to avoid predators such as water striders, which can efficiently locomote on the top side of the interface and skate around. And a particular collective behavior on the top side is the ant rafts, which I mentioned earlier, a semi-entangled system of hydrophobic terrestrial ants that links their arms together in a 2D shape and float in case of flooding so they can migrate to the nearest land. Where the worm buoy appears to be a brand new collective behavior on the bottom side of the interface, like mirror images of the ant rafts. Now, blackworms can breathe through their skins, but they can also supplement using their tails to obtain oxygen directly from air, like a snorkel, as seen there. Now, those pulsations of the dorsal blood vessel originate from the tail tip, and that's how they circulate their blood system. Now, the history of the tail behavior goes all the way back to the 18th century when Italian Catholic priest and biologist Lazzaro Spallanzani observed a worm with its tail protruding out of the water, and he called it the boat worm, or lumbrico abatello, as it resembles a boat. And it wasn't until the 1990s when Charlie explored this behavior more in depth, focusing on its neurobehavioral mechanism, as worms have an escape response when they sense shadows or vibrations when their tails are exposed on the top. See how quickly they yank their tails out, and this is in real time. Throughout all of these observations, the common theme is oxygenation. So why do they even need to supplement oxygenation in the first place? In an integrative comparative biology, or ICB, we published 2 years ago, we monitored dissolved oxygen in a 50 milliliter vial with 200 worms using an oxygen sensor. And this is part of aim 1 of my work on the biotic section. It took around 15 minutes to reach hypoxia and 30 minutes to reach anoxia. and that represents low dissolved oxygen conditions. This shows that a group of worms can collectively drive very low oxygenation in their environment. Notice here that the worm started out in a solid shape, solid spherical shape, and has now turned into a liquefied disentangled version. That's because worms disentangle and wave their tails for oxygenation, which emergently weakens their entanglement. So in a soft matter publication with Dr. Savoy, who was a grad student with Professor Goldman, and with the help of Ishant, we used a force sensor and found that the maximum entanglement force is in high oxygen is 3 times higher than in low oxygen. And we can actually pick up the blob using a 3D printed serrated end piece as shown here, which was suggested by Professor Goldman in this experiment called Blob on a Stick. So, and overall, these results suggest that entanglement can be driven by oxygenation. And of note, worms typically live in low oxygenation conditions in their environment, which is the benthic region, meaning that they actually prefer low dissolved oxygen condition and are stressed when you put them in high dissolved oxygen conditions. And this is part of aim 2 of my work, which is part of the physics of entanglement. So with the buoy, we place 100 worms and monitor the oxygenation with and without the buoy. And the no buoy condition was created by lightly tapping on the edge, which would scare the worms, put their tails down. We also counted the number of tails on the interface as shown here using Elastic, which is a machine learning software. And the buoy can sometimes accidentally fall, making this experiment sometimes difficult to run. And we ran 7 trials of each. The first curve you see here is for the no buoy case, starting from normoxic conditions or saturated oxygenation. Observe that the curve goes down to pretty much 0 after the experiment, replicating the same experiment I was showing earlier. For the with buoy case, observe a similar trend. However, it eventually pivots and starts to go up, reaching a new steady state value. And what that represents is that the worms have switched their respiration mode from absorbing directly from the water column, which is the water, and now have switched it to absorbing directly from the air using their tails. And speaking of the tails, this is the tail count on the interface with the buoy. And there are 2 key points here. The first one is the tail only comes up below hypoxia, as seen here, which allows for the formation of buoy and eventually stabilizes dissolved oxygen. The maximum tails indicate that not all of the worms are participating in the latching. And here you can see it at around 75% when the buoy is still stable and up there. Yet the entire structure is floating. That indicates that one tail can support more than its own body weight. So if a worm tail can support more than its own body weight, how large can these buoys get? So scaling up to the tens of thousands of worms, which is pretty much the entire culture we have in the lab. In this experiment, I initially filled a rectangular container very high with a water, with a height that the worms cannot reach the surface. And after 2 hours, I pumped it down to a lower level where the worms can now put their tails on the interface. This is the top view of the experiment. At a very high level of water, the worms tend to spread out as they reach up for oxygenation. Pumping down the water level so they can reach, observe the emergent formation of the buoy, clustering together and floating. And I added a little bit of food to drive locomotion as well. And from the side view, observe the arcing as they float collectively as one, observing that surface tension is supporting this entire structure. So can we find these buoys in nature? We traveled to Gulf Point State Park in Iowa to observe and collect worms, as these worms live along the edges of the freshwater pond through temperate regions of North America and Europe. And this location was actually in Charlie's paper, so we visited that site. They are typically covered in foliage year-round, and that foliage is actually a combination of algae, watermeal, and duckweed. And duckweed is a floating plant that has a little tiny leaf on the top and a root that is sticking down on the water, which I'll show later what that represents. And during this field study, we were unable to observe any worms, as most probably because worms pulled their tails down as soon as we approach them because they are sensitive to vibrations and shadows. Instead, we dredged some mud and collected a few worms and placed them in a container. And after a few hours, the wild worms did indeed replicate the same behavior. And on the right shows lab conditions using the same wild worms. What this denote is that this behavior doesn't depend on where the worms are reared, either they're wild or they're from farms, nor the type of water we're putting into it, but maybe more on the substrate or how many worms we're packing into a single container, again suggesting that this is an oxygen-driven formation. Overall, these results suggest that the function of the buoy allows worms to pack together tightly without being oxygen limited. Higher populations allow worms to locomote, work together, and even transition into a different stratum of their environment, suspending themselves upwards, as seen here, maybe to avoid predators that are on the bottom with them on the mud, like leeches. And a great example is the carnivorous freshwater leech, which is shown here in this diagram, this picture. And this freshwater leech is called Helobdella. It has two suckers, an anterior and a posterior, and they have a proboscis, which is a little straw that they use to pierce through the worm and then suck its internals. These leeches are present in their natural environment, but is also present with the worms when we buy them from commercial sources as unwanted pests. In my first year of grad school, just a side story, I was placing them in a separate container because I wanted to play with them. But to my dismay, my PI told me to dispose of them, and he said, "What are you doing here? Are you planning on keeping them as pets?" Anyway, in my rebellious second year of grad school, I decided to play around with the leeches anyway and put a place of worm blob with the leech so I could observe how they interact out of curiosity, leading us to the next project. This story is part of aim one of my work in the biotic section. Of note, these are the duckweeds that I discussed earlier that has little roots that are sticking out, and this foreshadows a future project that we are working on. In this work, published a few months ago in the Integrated Comparative Biology, or ICB, with Wade and Kenny, who are both undergrads that were working with me at that time, we observed that the leech tries to pull out a single worm from a blob, from an entangled blob, and they're having this leech is having difficulty of it because this is a highly entangled blob, and the worm tends to escape by pulling on it to each other. And a similar problem we all encounter in our lives is like pulling a single wire from a entangled mess. So this cohesive entanglement the worms showcases how the collective structure acts as a defense mechanism, resisting external forces and making it very difficult to extract a single prey by a predator. So here we see the leech successfully isolating a single worm, performing what I've called a spiral entombment, which is where the worm ventrally folds and creating a cavity in there to trap a worm. And this is actually a logarithmic spiral in mathematics. This entombment is necessary because blackworms are notorious for their ultra-fast escape swimming, as we've seen in the topology project. And this movement here allows the leech to trap a worm so that it can insert its proboscis. So after isolating a worm, the leech inserts its proboscis and proceeds to suck out its internal of the worm, leaving it as an empty husk at the end of this predation. In collaboration with Hiteshri and Pratusha, Hiteshri being an undergrad in our lab, and again, Pratusha is a research scientist, this project has continued on to investigate another common predator, which is planarians or flatworms, where we'll attempt to model them as active ribbons in the future. This has implications in complex biopolymers, and this work will be presented in the upcoming SICB conference, or at least the predator-prey biology part of it. Overall, these results highlight that entangled collective behavior has some level of protection against predation, and as well as the buoy where they move to a different stratum. Now let's go back to the worm buoy and delve into the mechanics and how these worms can hang on the interface freely together. Here's a magnified view of the tails from above, and notice those striations and pockets of light being reflected from due to their exposed skin or curvature of the water. And let's zoom into one of the tails and develop a theoretical model. So this is a magnified view of the tail from above, and notice that the worm has both longitudinal and circular muscles as seen here. Although we will be assuming a smooth cylinder for this approach, let's pull up a side view and observe that the leading segment of the worm creates this concave position, so this downward curved shape there, indicating that surface tension is providing load onto the tail. So I assumed a smooth, horizontally floating cylinder on the interface with radius r, defining a few parameters. This is the triple point where all three phases are in contact with one another, solid being the cylinder or the worm, liquid, which is water, and then gas being air. Phi is the wetting angle with respect to that, as well as the water curvature angle, where theta, which is tangential to that, is the equilibrium contact angle, which we assume to be constant as it is a material property here, where the angles are all related to each other by this identity. And here you can see that we only need to define 2 of 3 terms to fully define it. To understand how a worm can hang freely on the interface without sinking, we need to consider a force balance acting on its tail. So superimposing again from the backside of the worm tail with this geometry. And here you can see that the superimposition of this geometry reflects closely of how it looks like. There are three terms to consider: surface tension, gravity, and buoyancy forces. And the net force per unit length can be simplified to this equation here, where the bond number, which is the ratio of gravitational over capillary forces, is much, much less than 1, allowing us to neglect this second term and assume that the net force is primarily governed by surface tension forces. I made an interactive model in GeoGebra if anyone is interested in playing with a model where you could drag this red dot here to observe how the curvature is going and also the forces. Using image analysis, I estimated the wetting angle, which is around 140 degrees, and also the contact angle, which is around 70 degrees here, indicating that the worm is hydrophilic, which makes sense for a worm that is trying to keep itself wetted and not to dry out. Now, using the simplified equation, I calculated the theoretical net force using different contact angles from a wetting angle that is fully surfaced, meaning 0 degrees, to fully submerged, which is 180 degrees. Of note, the worm weighs around 7 milligrams in air, and this shaded region is the net force that's calculated for the geometry of the worm. And at 180 degrees, which is what the red dot here is showing, this is what the interface looks like with the cylinder here. That means that as soon as the cylinder or the worm touches the interface, the water curvature immediately becomes curved like this as the skin de-wets, and then slowly it will come down until it matches at least until it reaches an equilibrium with the hanging load of the worm here. So I believe this shows us a rough estimate of how much load surface tension is supporting with each tail within the correct order of magnitude. Of note, it seems like to carry more load like or other worms, each worms would just need to submerge by a little bit more, and that would allow the tails to support more load. Perhaps this is how they can hold such a huge amount of collective as shown in the previous video. Worms can also adjust how many segments they impose you by providing in order to provide more force, but they have to be careful or they could end up stuck on the interface like this poor worm over here, which is subjected to the bubbler. And of note, this worm is unable to come down if you just watch it for a few hours. With that, I presented the worm buoy, an entangled collective of blackworms that seems to defy gravity by floating on the air and water interface, perhaps for collective survival. We are currently modifying our model to closely reflect the segmentation islands we see of the tails. Worms seems to be capable of, again, adjusting their amount of segments they wish to present, or they could also expand and contract, which increases or decreases their wetting perimeter. And they do this, it's like an accordion once they latch up there. Someday, maybe we could use these principles to transport potentially heavy objects or an object that needs to be concealed using the underside of the interface. Speaking of that, let's discuss potential applications. I've already discussed that worms are capable of picking up objects. As seen here, there are actually food pellets that the worms are treadmilling from bottom to top. I was part of an international robotics paper earlier where we showcased that by integrating what we've learned from the worm blobs, maybe one day we can create soft robots enabling controls for tasks like grasping, collectively entangling with one another, collectively disentangling, moving around by itself as using through a random walk, collective locomotion, and that's chemotaxis on the top left corner. And we can also tune the entanglement strength here as shown in this blob on a stick project, as well as the pneumatic force you're applying there. Collective transport, so this robot here is actually transporting a piece of head, and these worms here are actually yanking on a piece of acrylic mesh. They're also capable of sorting things. Here, the worm blobs are actually selectively picking the green side of a traditional kitchen sponge. And I believe that's because due to the porosity of the kitchen sponge here, and where the green side has bigger holes. Um, they can also stitch materials together, like here you see acrylic mesh that the worms are seemingly stitching together like a living thread. And of course, this has biomedical approaches as well, or biomedical applications. Maybe they can also do structure assembly, like creating structures, or maybe strengthening damaged ones for search and rescue as well. And I do want to highlight that worms can also roll around for efficient locomotion, a rule we have yet to decompose, but it certainly is directly applicable to entangled swarm robots. Worms can use their environment around them as well. Here is the duckweed that I was discussing earlier. Worms are capable of using the roots from floating plants to suspend themselves collectively without having to expose their tails on the interface. Here's a fancy photo of that. In soft robotics, the main objective is to create compliant systems, meaning flexible and adaptive, for diverse applications such as um, grasping delicate or something that has complex geometry, as seen there. Unfortunately, though, current designs rely on expensive sensors for precision and are tethered on one end, as you see in here, for actuation such as pneumatics or hydraulics, or that's where their power sources. Soft gripper technology, originally designed for handling delicate samples in deep sea, hints at a future where adaptive, untethered soft robots could transform how we interact with complex environments. In conclusion, culminating over 4 years of research, I uncovered how worm blobs use entanglement to adapt and survive in dynamic environments. From their topological coordination and controlling their strengths um, of entanglement to material gathering and how they collectively exploit surface tension. I hope these insights lay the groundwork for envisioning untethered, soft, entangled swarm robots capable of autonomously wrapping around, transporting, protecting delicate structures, or maybe even shape shifting into one. Such technology could transform search and rescue operations, deep sea and space exploration, architecture, and more, showcasing the potential for nature inspired entangled robots for real world challenges. So, this concludes my thesis, and I will take any questions now.